Next week, so just to start off, this is the last of the boring lectures. They haven't been super exciting, I think, up to this point because they've just been science-y. But I think once we get these foundations, when we start talking about learning, when we start talking about circuits in the brain, when we start talking about functional things like hormones and sympathetics, it lets us talk about real stuff that we do.
These lectures have not been exciting.
Okay. Well, hopefully they're going to be fun, more fun, once we can start talking about real stuff. But I want to correct myself. I lied to you all last week. Yeah, and so I'm probably going to lie to you more, but just to clarify that, it's not the Overkalic study. Absolutely, you guys are right. But I mean, half of you already knew that. It's the Dutch hunger famine. And so just a quickie review of the first 3 lectures. The first lecture is about thinking functionally. Perfect. People are actually coming. So yeah, just file in. There's going to be a lot of people who don't know how to get here because some people have emailed me that are not in the department that wanted to come, and I think that they think it's going to be in that room. So I didn't put a sign up, so I don't know if anybody's over there or if in like. Were there really?
The psychology friend? There were a lot of There were about 7 or 8.
Yeah, so I don't know if it's somebody is willing in like a few minutes to run over there and just see if anybody's there and bring them back.
That's closest to the door.
And it was very fast. And fleet of foot.
Yeah, if you don't mind. Thank you.
Did you see them in the library? Just see if anyone was hanging around the library or the residence.
Is there something going on in the library?
Yeah, there's another meeting for psychology residents.
So I should go into that one and say.
No, just see if anyone was hanging around. It would be fun. Just go into it, yell fire. Nice to
Thank you. I appreciate it. So again, it's just to review, the first lecture was on thinking functionally and not reductionistically. I'm going to say that a whole bunch. We want to think functionally. We want to think about this in a practical way that we can understand. Then we talked about genetics, and we're going to go over a little bit of that when we talk about the DNA in the nucleus of a neuron. Then we talked about epigenetics, which is where the Dutch hunger famine study came in. It was during World War II. There was a period of about 3 months where there wasn't a lot of nutrients, and mothers who were in their 3rd trimester had babies that then had lots of difficulties handling glucose. And that didn't happen for mothers who were, say, in their 1st trimester or babies who were just 3 months old. So there's this critical period when your DNA is sort of setting the stage epigenetically for how it's going to respond to certain environmental signals over a lifespan. And so that's sort of what we were talking about with epigenetics. And now we're going to talk about the neuron. So this is going to be where we're getting sort of to the functional basis of the nervous system. And again, like I had said, this is the end of sort of the basic science that's really hard to apply to phenomenon. We can't really see phenomenon on a DNA level. We can't see phenomenon on an epigenetic level. We can't really even see phenomenon on an individual neuron level that mean anything to us. In the next step, we can see real phenomena, so things that we can actually observe happening in a person. We're going to talk about endocrine responses. What does it mean when you get, thyroid hormone or cortisol? And then we're going to talk about, functional pathways. So what happens when a certain pathway gets activated, and what sort of behaviors would that translate to? So when we get started, just going to cover a few things that are going to let us build on this as we move forward. So I'm just going to start with a review that most of you already know. So the brain is made up of neurons, and the brain is part of the central nervous system. So pretty much the brain, the brainstem, the spine, and the glia make up what we call the CNS. And so we're just going to do a very quick overview because we're going to build on this as we go, and we're going to spend a whole lecture on neuroanatomy, which is I think, going to be a fun lecture. So here's the frontal area of the brain. I like to think of it as sort of roughly split into 2 parts, the prefrontal and then the frontal where we have our motor cortex and our premotor cortex. And this is going to control planning to some extent, particularly with movement, but with other things as well. Here we have audience interaction. What's the name of this lobe here? The parietal lobe. And what does it do? Roughly. So what's this strip up in here? So this is our sensory cortex. So this is going to take in the information. So this is going to get where all of our sensory inputs are translated. And this one back here, occipital, and this is where we do vision, right? And here's the temporal, good. And this is where we're going to sort of do some memory. We're going to do some auditory learning stuff. This down here, the cerebellum. And the cerebellum has a ton of neurons in it. And it's what we do to sort of correct our motor movements, which you can imagine is pretty important. So, we throw a basketball, it's too far to the left, we correct to the right. We extend one way, we need a balance. So that's all that's happening there. Then we get the limbic system, which we're going to be very concerned of, that's sort of buried inside of here. And interestingly, I'm going to make the argument that this part of the brain is limbic. So I want to think about this, you all to think about this more as we move forward as limbic. It's more limbic than it is frontal is what I'll say. And those connections go more this way than they go this way. And this is a cool area of the brain. And the peripheral nervous system is the stuff outside that is going to take the action neurons from the spinal cord to our muscles to let us move. And then it's going to sense stuff in our environment. So pain, temperature, our gut, what it's doing, and send that information back up into here largely. So that's just a rough sense there. Now we'll talk a little bit about the neuron. So to talk about the neuron, we have to talk about Santiago Ramon y Cajal, which until like a week ago or 2 weeks ago, I thought was two people. He's got an E in his name. It's one guy. I did not know that, but no, that's not him. I'll tell you about that guy in just a minute. I don't know if you guys may know this guy, but so he's the guy who sort of described the functional unit of the brain. We thought the brain was a mush. He comes in and says, no, it's not really a mush. There are functional discrete units here called neurons. And he stained them with Golgi staining, I think, and these beautiful pictures of neurons started creating them, and they were pretty beautiful. So what the neuron does is it sort of can receive input and it can send a signal if it wants to. There's also glia, which we're not going to talk a whole bunch about, but really probably are very important. And so there's astrocytes, which are the most numerous. There's oligodendrocytes or Schwann cells, which are going to do our myelination and stuff like that. There's stuff that's going to monitor the immune system in the brain. And it's important for that to be healthy. There's some data that's suggesting some psychiatric illness is related to malfunctions in the glia. And I think that we're going to find that it's more and more important as we go on. Although probably, it's not going to explain everything. Nothing's going to explain everything. But it's a part of this puzzle. So we have a lot of neurons. Anybody want to venture a guess at how many neurons we have in our brain? Okay, so that's a good question. So it doesn't change a whole bunch in a sense of neurons. Our neurons stay relatively the same. Our synapses change a whole bunch. But say at an adult age, how many neurons do we have? Unless you're talking about like at 1 month prenatally. But what about us, more or less? So very good guess. I mean, within an order of magnitude. So we have 100 billion neurons in our brain. And somewhere between 100 trillion to 500 trillion synapses. And these are really, really huge numbers. So just to give you an idea, a fly, want to guess how many neurons a fly has in its central nervous system? 100,000, okay? And so we have 1 million times as many neurons. And a fly can do some pretty complicated stuff. It can understand social patterns, flying, reproduction, feeding. I mean, a fly has a pretty large repertoire of behaviors. And so, again, I'm not saying that we're going to be able to really understand on a neuronal level how to go one-to-one from this neuronal pattern to understanding this behavior. But I think that we're going to be able to zoom in on some areas and try and understand some behavior based on that level. So this is Sean Carbonell. He was a student here. He goes by the name of Big Axon, which I think is hilarious. And he got a tattoo of a neuron on his arm. He owns a company now called OncoSynergy. He's just an interesting guy. I don't know anyone else who has a... he just got married. He's a good guy, though. And so here's the stereotypic neuron here. And all that we're going to take away here is we got the dendrites on one end. The sort of in here is the soma, which is Latin or some old language for body, and the nucleus in there. And then the axon with sort of the axonal projections. And just to get a sense of the relative size of these things. Axons can get really, really long, potentially like our motor axons from, say, our spinal cord to our toes. So if our hand was the size of the cell body, the axon could be about a mile long, just to give you sort of a relative sort of difference. Most axons aren't relatively that long, but compared to the size of the body, they're all pretty long. Most of them are just bouncing, maybe a third or a fourth of that distance. And then again, a sense of size. The soma is around 4 to 100 microns, which again, if we're starting to think about these things zooming in and out, you can think of protons being, different sizes, but on the order of microns. And so, a protein in there is a sizable element. So it's not, on that level, it's not microscopic. On that level, it's very much a structural, and we're going to see that proteins are the structure of this, and so it makes sense for it to have a size within that scale. So here's a more fancy picture of a neuron, and all that I want you all to sort of notice here is that there are dendrites, and they're sort of very branched. We have axons coming in from another cell, and then here we have the synapse. We're going to talk a good bit about the synapse. You know all about this sort of stuff. We've got the cell body with a nucleus and all the machinery that we need. All of our genetics and epigenetics happens in here. And then we've got the axon hillock, which is the area of the most excitation electronically. And then we have our axon, and then more sort of synapses out here. And just notice that synapses can be in both, usually they're on the dendrite, but they can also be on the soma, and they can be on the axon. And so there was this guy named Dale who was sort of this big neuroscientist, and he had two big laws, and they were pretty helpful at the time to understand how this works. And his first law was that every single action potential that got fired propagated to every single axon terminal. And he said that every single axon terminal within a single neuron only releases a single neurotransmitter. Both of those ain't true, unfortunately. So for a lot of them, that is true, but it's just interesting to sort of see the different mechanisms of modulation that we can have, and sometimes it's these nuances that allow some of the finer appreciations of things to happen. So again, we can think about three different types of neurons generally. So there's the afferent, which are going to bring the information back, the efferent, which is sort of sending the motor signals out, and then the interneurons, which is really just neuron to neuron, and that's going to be, for us, the most important neurons generally, because that's what's doing the modulation in our brain. So if it starts in the brain and ends in the brain, it's pretty much an interneuron. So again, like we had said, the neurotransmitters have to get made here, right? So we've got transporters to get our amino acid precursors in here. In here in our DNA, we're going to have our enzymes, right, which are proteins. So all this regulation happens here. We make our enzymes, we create our neurotransmitters, but it doesn't do us a lot of good here, right? So something has to happen to get it down here. Because again, I want you all to think functionally. The same way that you make the cars in the Nissan plant, you got to ship them. You've got to have a mechanism for this. It's not magic. We got to sort of think about what's really going on here. And there is a transporter. And look how awesome this is. Like, this stuff, so you bind your neurotransmitter here. And this walks across a microtubule. And literally, based on conformational changes by breaking phosphate bonds, you are creating rotational changes that are walking this. And this is literally what happens. Like, how cool is that? So,
forward versus backwards?
So again, I would have to go back and review all of this in some detail to give you a really fair answer of how this is happening. My guess is that it's simply the order of phosphorylation and dephosphorylation of these things. So it's got two legs of it. A phosphorylation of this probably causes it. And this is just me conjecturing. Counterclockwise rotation here, that's going to change the conformation to make this binding site more exposed. Again, these are just me making stuff up, but it's something probably... Perfect. That absolutely. I'm sure there's a science behind this.
It's important to remember, guys, too, that the small neurotransmitters are actually made in the presynaptic terminal. It's these big chunks, these peptides.
That's right.
Made in the cell body and shipped down these microtubules.
And that's a good distinction. And we'll get there. Sorry, we'll get there when we talk about different kinds of neurotransmitters. But that's true, and we'll even get in more detail of what's happening at the sort of synaptic terminal in regards to all sorts of things like packaging and all that. So I love this picture. It doesn't have a lot of emotional meaning to you all. So I was in high school chemistry class. That's right. I was in high school chemistry class, and I did this lab, and there was an explanation section. And so I just posted this picture and then skipped right on to the next part of the lab. And so I did it with Andy's brother, John, who's a radiologist here. He's a really smart guy. And so we turned it in. And we both turned in the exact same lab. I get an A, and he gets a B. And again, what was going on, I didn't go to the best high school, admittedly, is the teacher had a assumption going into it that I was an A student for some reason and this kid was a B student, and was just thinking again in this categorical way and not looking at what's actually in front of her and the fact that it's the same thing. And we actually asked her about it, and she gave us some bogus answer. But anyways, I think he ended up getting an A as well. So we're going to talk very briefly through the whole process of what a neurotransmitter does to communicate, and then we're going to really focus in on it and hammer it down. So we'll start with a neurotransmitter being released from the presynaptic neuron. It gets into the cleft, and then it sort of diffuses across, and it binds to receptors. And now we're going to talk about two kinds of receptors, and we're going to talk about receptors in ridiculous amounts of detail. So we have ionotropic receptors, and we have metabotropic receptors. And these are also known as type 1 and type 2 receptors. And so again, I want you to think about this very functionally. A ligand, which just means that it can bind, that's what ligating is, comes in and binds to this ionotropic receptor. And again, you can think about this in lock and key, and we'll get to that eventually. But that ligand is some molecule that has some functional part that can bind to this, which causes a conformational change in a protein. Now again, think about this. This is literally what happens. It is a tube. It changes the conformation of this, and it allows a passage which allows these things to go through. This is pretty realistically what's happening, and so we can sort of understand that on a physical level. So this is very fast how it happens, and it creates an ion difference across the membrane. This is a metabotropic receptor here, and you can see that it is not directly connected to an ion channel, but what it does is it influences some second messenger, and sometimes it takes many, many steps, but ultimately it can either create an ion channel to open, or it can do something that's ultimately going to influence DNA. And this is often how we see long-term effects of neurons. And again, so something is happening in here that's activating a transcription factor, that's opening up the histones, which is allowing a transcription site to be bound, which is changing the modifications of the protein when it's done. And it can actually create receptors to then go back into the membrane, or it can create whatever it wants to do to change that individual neuron. So now we're going to talk a little bit about the action potential. And so I would just talk through this whole case on the board, if that's okay, because it's just easier. And so we'll go through the whole thing again very quickly. I think I've got room here. All right. So we've got these dendrites out here, and I'm going to exaggerate one of them. That's going to come onto our neuron, and it's got other dendrites out here that are branching. And, okay, here's our dendrites. Whatever. I'm not a great artist, but this will suffice. All right. So here's our presynaptic neuron, and it's going to release neurotransmitters. Okay? So we've got receptors here. All right, we're going to draw two different kinds of receptors. All right, so this will be an ionotropic receptor, and this will be a metabotropic receptor. So there is an electric potential inside this neuron, and as you can see there, it's negative. So there's more negative in here, and there's more positive out here. And we can talk about how that happens. It's sort of sodium, potassium, ATPase pumps, and all that good stuff. So again, think about this just like a physical thing that you could touch. It's all closed off, and there's a phospholipid bilayer, which is not going to allow these charges to just go across. So this binds to this, all right? It creates a conformational change. And so what it does is it takes this thing that's closed and it opens it up. And so now we've got a passage for these ions here to get inside. And so we get a little bit of plus here, and it starts diffusing in here. So that's the first step, an increase in, or a decrease in negative charge inside the neuron. On this area here called the axon hillock, there's ion receptors that are voltage gated. Okay, so these are ligand gated, meaning that it needs a neurotransmitter to bind to it to open up to allow this in. This is voltage gated, which just means once a certain voltage is reached, it opens. And they're most densely packed at the axon hillock. And what you can see there is it goes up slowly as you get more and more of these ion channels opening up. And then once it reaches a particular threshold, which is around negative 50 millivolts, millielectrovolts or whatever, some of these start opening. And then you just start get some rushing in. And you can imagine how this could be sort of an accelerating cascade, because once one opens up, it creates a less negative neuron, which will open up more, and so increasingly less negative. And this gets propagated down here. And that's how an action potential works. It's, again, a very physical phenomenon of something coming in, a protein creating a real change, allowing ions in, opening up these voltage-gated channels, propagating an action potential down here. And so now we're going to make another synapse here, so we can see sort of this recreated. So here we have synaptic vesicles, and I have a funny story in a little bit. I'll just tell you now. We were learning about this in medical school, and I raised my hand, and I had a dumb question probably, but I asked, actually what I asked is, can the spermatic vesicles have more than one neurotransmitter in it? And I obviously felt really dumb, and the whole class laughed because I called them spermatic vesicles. But I remembered that, and I won't forget that. And I remembered it probably because I got some epinephrine, I got some cortisol, and I created a situation that's very likely to code for learning. And we're going to get to that next time. And why do we remember what we remember, and how does that shape us? Which is very, very important in psych, because we learn. We're learning creatures, very much so. So again, this comes down here. You have voltage-gated channels down here, but as opposed to allowing in sodium, what they do is they allow in calcium. So calcium comes in, and what you very much, again, very physically get is the calcium is creating a conformational change through a cascade that is going to pull these vesicles, and I can't draw very well at all, but you can imagine this vesicle getting pulled down, and then in the next phase it sort of looks like this, and then it's got these things in it. And so it gets pulled into this, it becomes part of the phospholipid bilayer, and then these neurotransmitters can come out and bind to the next part. Okay, so this is a very physical phenomenon. There's nothing magical happening here, and this is a way that we can understand it.
Like the balloon is blue?
Like the balloon is blue. Again, and the reason that I like thinking about it this way is because it allows us to view a system in a realistic way. So if we can see where it's broken, we can then think about different ways to fix it. And I really do think that that is what we're approaching to some extent in psychiatry. So again, now to take this to a sort of a functional level, let's just start at the top in something that we do understand, which is motor actions. Okay, so our prefrontal cortex takes in some input. It, for some reason, wants to have a coordinated muscle movement. So our prefrontal cortex plans it. It plans it in a very realistic sense, like I want to reach out and grab something. That I want to reach out and grab something is coded by an algorithm that sends different signals to different areas of our motor cortex. It's prefrontal cortex, pyramidal system, extrapyramidal system is all coordinating this movement. So we're already talking about multiple neurons, but let's break it down to an individual neuron and say signal goes into the motor to do something. Action potential happens in the motor cortex, goes down based on our homunculus or whatever. It's more complicated than that, but goes down to our spinal cord to what's going to ultimately synapse with that target muscle. Synapses in the spinal cord, if it's a strong enough signal, It then sends it out to the muscle. So we've got neuron to neuron to neuron, ultimately neuron to muscle, and this is where the action is going to happen. So again, it's not magic what happens. Acetylcholine gets released from the neuron. It binds to the muscle cell. Once it binds to the muscle cell, a lot of stuff happens. It's going to open up some sites, ultimately, on a muscle fiber. That's going to allow actin and all of those things that we learned about and don't want to remember about, the sarcomeres and all that stuff. What you end up getting is titin, which is actually, this is the biggest protein in the body. Just an interesting fact. It can be, I think, like 1,000, up to 1,000, something, whatever, 1,000 something. I don't remember. I don't want to say whether it was microns or whether it was 1,000 nanometers, one or the other. But what it's literally doing is creating a conformational change of a pulling together or a pulling apart. And so you can see here how that conformational change of a protein over a mass layer, again, we're sending a coordinated signal of, contracting one set of muscles, relaxing another set of muscles. The cerebellum is making sure we balance when we do this stuff. So it's a coordinated effort. But at the end of the day, it's a very real physical phenomenon that's happening. A lot of stuff is coming in to create a very fluid, purposeful movement. And what case I am going to make, and I think that it's very useful to think about that this way, is that emotions are the opposite of this. We get this input of sensory information coming in, and we're taking it in from everywhere. It's pain, it's our cognition in our environment, it's how relaxed we are, it's our heart rate, it's the way that our muscles are flexed. We take in all of this environment, and it gets filtered in a similar way as our vision does, in different levels to produce a finely tuned emotion. And I think it's, again, very much happening on a very physical, real way. We have ways of taking in this input. We have interneurons of relating it. And that can send sort of a coordinated signal that can be then processed as, liking or loving. And there's different kinds of love. In the same way that we don't think of movements as sort of these, extend this, flex this. When we play the piano, that's not what we're thinking. We're thinking of the playing. So it's sort of this purposeful thing. And I think in reverse, emotions are that. And what I think that lets us do in a very cool way is sort of get into the system, right? So here's an interesting thing. I want you all to just put a huge grin on your face. And then sort of relax. And you sort of feel happy. And we know that. We know that this happens. Your body language and what you do influences your emotions. And think about why that might be. Because usually when you're smiling, it's unconscious and it's linked with other things. It's pushing our brain into interpreting this stimulus. We're just tricking our brain to some extent into going into you must be smiling. We're getting these motor signals here which are linked to happy signals. And again, I'm talking in a way that we don't quite understand yet, but my guess is that's what's happening. And as we begin to understand things on this level, we can start manipulating things on this level, which is very cool, I think. Does that sound sort of reasonable to everybody? The idea of, somebody can feel anxious because their heart is racing. But if they're, cognitively aware of it, that it's normal for their heart to be racing in that situation, they can turn that emotion down. So it's regulated on many levels, but very much what we're getting in from our environment is largely determining these things. And so again, to just use this car analogy, here's a wonderful commercial, and I'll just play it, and then we can talk about it. It's a couple of minutes long, but I think it's worthwhile. So the idea behind this is, one, to buy a Honda, and secondly, to sort of see how there are a lot of steps that had to make that happen. And if you just look at them as their individual pieces, you're really missing the big picture. Each of those steps was guided by their own physical principles. And a break in any single one of those steps could have broken anything in the system later. And you could imagine multiple pathways leading in, multiple bottlenecks. And so if you just look at one piece of that, you're really missing the big picture of what's going on. And it's also showing that that was pretty complicated, right? And when things work, we don't have to worry about it. But when things are broken, if that wasn't working for some reason, like your neurons aren't firing right, it's helpful to have an understanding of the physics, of what's going on, of why this is not working, so you can then go in and in a reasonable way fix it. So if one part's not working, if you can find a way in the system right after that, you might be able to get the sort of most direct path to correct the system. So we'll focus a bit on neurotransmitters. And so there's a few rules for things to be neurotransmitters. It's got to be made in the neuron. It's got to affect the postsynaptic neuron. It's got to have a way of being communicated after an action potential goes off and all that sort of thing. These are the different kinds of neurotransmitters. There's the amino acid neurotransmitters like cycloserine and stuff like that we're hearing some about. The monoamines, which are the ones that we're very familiar with. These are our serotonin, histamine sort of stuff. The peptides, and peptides are small proteins, pretty much is the easiest way to think of them. The definition of a peptide is just less than 50 proteins. So it's a protein is generally more than 50 amino acids. A peptide is generally less than 50 amino acids. And then there's small molecules, like really small stuff, like nitric oxide, which we're finding is super important. And nitric oxide can go backwards and all sorts of crazy stuff. All right, so the idea here is that there's only a few hundred, maybe, something in the order of 100 to a few hundred, but it lets us do a lot of different things. And so you can think about neurotransmitters to some extent like the letters of the alphabet, in the sense that, if you can put them in an organized way, you can create a lot of different messages. So before I move on, I want to talk about other forms of communication. So we'll start with cell-to-cell. And what a cell-to-cell communication would be like passing a note. Okay, so I physically walk up to Ivan and I pass him a note. Do you like me? Check yes or no. He sees it. It happens quickly, but no one else sees it, and it requires direct contact. The next is paracrine. I come over and I whisper, psh, 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 psh. A small group hears it. It requires direct contact, but no one else further away hears it. The third is neurotransmitters, and this is what neurons use. And so this is akin to a text message. All right, I can send a text message to Carrie, and I can go right by Peter, but only Carrie and I know what's going on. It's fast, it's sort of direct, and it's can span long distances without touching things in between. And then there's endocrine, when I send you all my spam emails, right? So it goes to only the people that I want to go to, and it allows us all to coordinate. 4 p.m., be here, show up like this, be ready to learn. And so these are the different ways that we have of communicating. And so an example of why that would be important, say in the endocrine system, is, you get stressed, you've got to do a lot of coordinated signals very quickly to respond well. So you want an endocrine signal to go across, it gets in the bloodstream, and it goes, hey, do you respond to cortisol? Hey, do you respond to cortisol? And if they do, they can have different effects. So it can allow these cascades of effects. That's what we're going to talk about next week, is the endocrine system. And I think that's going to be a fun lecture, because it starts getting very practical, and you'll start seeing, hey, this happens to me. Hey, this is really what's going on inside of my body, where it's hard for us to think about neurotransmitters, but the goal is going to be we're going to connect these things because they're obviously connected. It's our brain that controls our endocrine system, so it's these neurotransmitters that decide yes, go, and if you go, how much, and shut off when, and those sorts of things. So we've got to understand this first, because if there's a problem there, there's obviously a problem afterwards. But once we start getting more systems, it starts letting us have more fun with this stuff. So now we'll talk a little bit about receptors. So what's a serotonin receptor? It's a protein. Yeah, that's a wonderful answer. What makes a serotonin receptor. Okay. And again, this is an answer, think of what I'm thinking. Why do we call a GABA receptor and a serotonin receptor a serotonin receptor? Because that's what they respond to. Okay, so we are naming the receptor based on what binds to it. So I want you all to think about a lock and key method here pretty much. So if serotonin is our key, it can go into a lot of different locks. And we're going to name all of those locks a serotonin lock. But I put it in one serotonin lock and I turn it and the TV turns on. I put it into another serotonin lock and I turn it and the lights go off. I put it into this serotonin receptor, I turn it, and Mandrill is even more awesome than he is now. And so, which is not even possible. This one doesn't even exist.
Come on.
So the idea is that these receptors do very, very, very different things. But we're just calling them all serotonin receptors because serotonin activates them. That's one of the biggest points that I want to get across. Serotonin receptors are very, very, very different. They have a site on them that allows serotonin to bind to them, but they also look different. So just because serotonin can bind to them, that does not mean that everything that can bind to a 5-HT1A receptor can bind to a 5-HT7 receptor. There is something there that's a serotonin binding site, which is not necessarily where our drugs are binding. So again, understand that it's sort of silly to just think of serotonin receptors as being the same thing, because they're very, very, very much not. And just to sort of show that in a sense of scale, I'll just click on this. And again, you can see, look at all these types of receptors. I mean, it's insanity, right? And they all do very, very, very different things. And again, I can go on with this stuff to get that point across. But I think that's a really, really important point. And next, we're going to focus on the GABA receptor, because I think this is another very useful thing to understand. So here's a cartoony GABA receptor, all right? It's made out of 5 different proteins to form this one heterodimer, because there's 5 parts to it. So 2 alphas, 2 betas, and a gamma, generally. It doesn't have to be that way. It cannot have a gamma or whatever. So GABA binds between the between the alpha and the beta site. So GABA can bind to all of these. It doesn't necessarily do the same thing to all of them, but it more or less opens the channel, not to the same extent necessarily, but opens this channel. Again, we're thinking about the same sort of conformational opening to allow chloride to enter a cell. So there's only 2 GABA receptors. There's a GABA A and a GABA B. That makes it a little bit more interesting. We're going to focus on GABA A alone, which is what we're considering this heterodimer complex that's generally in the brain. So all that GABA A can do, there's only one of them, so it can only do one thing, is open up and allow chloride to enter the cell. But benzodiazepines, or any other drugs that bind this, don't bind to the same GABA site. In fact, GABA sites don't exist, and benzodiazepines only exist in sort of retrospect. All that we're doing is there's this protein in here, and this molecule is coming in, and it happens to have a shape that allows it to bind to this receptor. Again, this was not pre-planned. Our body was not planned to have a benzodiazepine site. This is not a benzodiazepine site. This is just molecules that a benzodiazepine molecule can mess with. So again, it's a very much, these things are only done retrospectively. We give a medicine, it does this thing, and then we sort of can figure this out. But benzodiazepines can only bind to certain alpha gammas. So they can't bind to alpha 4 and alpha 6, and they can't bind to certain gammas. And different benzodiazepines bind differently to these things. And we have distributions of different alpha 1, alpha 3, beta 7, gamma, whatever, in different parts of our brain. And so I'm not exactly sure whether there's a different gene that codes for these, or if this is a post-translational modification. I'm not sure of that. But so you can imagine now, you have one individual, a single individual. I've got certain GABA molecules in certain parts of my brain, different GABA receptors in some parts of the brain, different GABA receptors in other parts of my brain. I take a single benzodiazepine. It might bind with some high affinity to some parts and not with such a high affinity to other parts. I take a different benzodiazepine and it may act very differently. And this is completely outside of half-life. This is completely outside of potency. This is just saying that certain parts of our brain are going to bind certain receptors differently. So, therefore, not all benzos are just benzos, and that makes sense. Some benzos might work well for a person. Some benzos might cause more sedation than anxiolytic properties. Some benzos might cause muscle relaxation. Some benzos might cause sleepiness, and it all depends what these things distributions are in our brain. And so even if, let's say, Ivan and I had the exact same distribution of GABA receptors in our brain, that does not mean that we necessarily have to have the same response to even the same benzo, right? Because what's lying under this is a neuron with genetics, with its own sorts of connections and strengths of connections and numbers of things. And, if my neuron produces some different gene than his neuron, we could still, even given the same receptors, have a different response. But the truth is that we have very different receptors. And so there is this complexity. Of course people can have different responses to the same medications because we have different distributions of these things. We have different underlying chemistries. And that sort of brings up a point. So this is just on the neuronal level. There are things that happen outside of the neuronal level. So anytime we take a drug, we don't know what's going to happen in our body, right? So that is a behavior. You take a drug. That is a stressor to some extent. What is this drug going to do to me? Is it going to hurt me? Is it going to help me? Outside of that drug effect, we have that stressor, which is going to create cortisol. It's going to create other things too. It's going to create cognitions maybe. Certain cognitions may be more so in some type of people. Certain amounts of cortisol more in maybe some type of people. Some people have side effects to every drug. That's not to say that they're lying, that they're faking. It just means that something is going on that's likely creating this. And my guess is that it's the stressor. It is, oh my gosh, I'm taking a medicine. What's going to happen to me? Stress, cortisol goes up. What happens when cortisol goes up? We get an upset stomach. Our heart races. We get nervous. We get uncomfortable. We get diarrhea. So if somebody has a side effect to every medication, it doesn't mean that they're even necessarily medication sensitive. It can just mean that they're anxious. But what I want you to think about is something real is happening. They're probably not lying to you. So we've got we can get through this. That's okay. I'll show you. Well, before I show you this, we'll talk again about a lock and key in a very real way, right? So what happens when I put a key and I put it in a lock? Nothing, right? So until I turn it, right? But when I turn it, what happens? Right, so again, this is a physical phenomenon. I'm putting something in here. There's normal forces. There's tensile strengths. There's a change in sort of a normal force of resistance as I'm going to open the door. There's a physical phenomenon that's stopping this door from being opened. Let's say I want to get into that room, but I don't have the key. How can I get in? I'm sorry? No, absolutely. I think what you said was correct. What can I do? Break in. How could I break in? Yeah, so there's once you understand the system, there's ways around the system. What did you do before you came into this residency, Joseph? Get out of here! Again, you could breach the lock. You could blow the lock out. You could say forget the lock. You could just cut a square in the door and create a door within the door. You could go around and jump through the window, and that's the sort of thinking that I want to try and use. This poor guy. All right. Again, they rolled with it. I think they did a great job given all that happened there. Okay. I'm going to run out of time. So before I run out of time, I want to talk about one thing. And that's, again, the idea that what we're dealing with here is more behaviors, and what we're calling diseases are really not diseases. And that's a really controversial sort of thing, to say depression's not a disease. It's a real thing, but it's not a disease in the etiologic sense, right? Cough isn't a disease, but it sucks. Certain things cause a cough. And we're going to talk about that a lot more. But I want to think about, what are the sort of things that can lead to illness behavior? And, because we talked about this a little bit last time, and I thought about this and tried to come up with, what can happen that we have to create illness behavior. And I came up with a few things. And the first one was just sort of like pathological learning, right? So this is everything is working fine, but we have an unfortunate pairing that does not allows us to create sort of a bad association. So we want to be able to learn. When we get scared, we want to be able to learn from that, recognize that that's a dangerous situation, and run away. When I see a lion, I get scared, I run away, I survive. I want to realize, yes, that lion was dangerous. I did a good thing by running away and surviving. F, for example, we get a bad association. For some reason, I get scared of Ivan's yellow tie because something horrible happens and I associate with that yellow tie. I run away and I survive and I learn something. All of our pathways are perfect on the sort of modulistic sense. I've just made a bad association. And this happens all the time. This is where a lot of our sort of irrational stuff comes from. A single learning experience that we then, or, I won't eat this because I had a bad experience, even though it probably wasn't that food that you ate, it was probably something else. We can all get into this sort of thing. Or, anyways, I won't go further on that one. So that is the second one. So I'm going to call this adaptive to maladaptive. So this is when you learn something that's very helpful to you in a certain environment. So say, I have to cry, I have to be overreactionary to get my mom to come to me. So I learn to do that, it works, it gets my attention, but as I get older, I don't learn to adapt to other situations, so that same behavior that was adaptive has now become maladaptive. Similarly, it's a person who sort of has a lot of stressors, they can't trust anybody, so they don't, so it protects them early on, but then when they're in a nurturing environment and they should trust, they can't. They can't unlearn that. So that's sort of an adaptive phenomenon that becomes a maladaptive phenomenon. Exactly. You can't shut it off. That sort of thing. The third one is what I'll call, and I think this is one of the biggest ones, homeostasis or catch-22s. And this is what I think is happening in most of our more subtle disorders. So if you think about it, we have lots of modulatory things in our brain. And we talked about this on every single level. We can upregulate the number of transmitters. We can upregulate the number of dendrites. We can have other ways to come into pathways to turn up or down certain things. And so our body does its best to try and create an environment that's stable for us. Sometimes these mechanisms can get overrun. So say Parkinson's disease, right? We can lose a lot of our dopamine before we start expressing these symptoms because we're compensating by some way. But at some point, your body can't keep up and illness gets expressed. And I think that this happens often. And, like we were talking about, we look at these genes and expect to find a gene. For these more subtle disorders, I think what we're going to find is this gene's only letting us compensate 80%. This epigenetics is only letting us do 60%. This learning has created this maladaptiveness 15%. This endocrine signal has gotten out of whack. And it's this culmination of things that are not allowing us to keep this homeostasis in a more stressful environment. So people have different adaptive abilities, and that's my guess of where this rubber meets the road. Some people have wonderful adaptive abilities. They can adapt to a lot. No stressor will throw them off. Some people have limited adaptive abilities. Lots of stressors can throw them off. Or there's this catch-22, which I think happens very commonly. So the world is a dangerous place. I need cortisol to be aware of it, but I pay a price for that cortisol. So I need to be awake, I need to be alert, but by doing that, I've got to suffer with some anxiety. That's just got to go along with it. You can't have one without the other. I need to stay awake because this stuff is going on. You're going to have this fatigue. So these things that to get one thing, you got to pay a price for another. And the fourth is, my guess, is just going to be a broken system. So, you just don't have something that is necessary by some insult. So something is necessary somewhere along the way, and you just don't have it. It's not there, and there's no way to get around it. And this is a more biologic sort of situation. And probably doesn't represent a lot of our illness, but maybe does to some extent. Maybe this can then feed into this. So you have a broken down system that is then going to ultimately lead to some maladaptive things happening. And that's sort of, oh, I'm sorry, and the last one, which is extremely important, and that Dr. Hobbs and I, and then I'll end on this, talked about was attractor phenomenon. And I'll just talk about 3 different possibilities. So one is, let's say, I'm a big guy, I'm overweight, I've got my supersized soda in this hand, I got my cigarette in this hand, I'm slurping it down. My adipose cells are just engorged with fat. I drink the soda, my blood sugar goes up, right? My pancreas says, oop, we got to get rid of that blood sugar, give out the insulin, let that body store it as fat. The adipose tissues say, nope, we're full, we're not taking any more, we're not storing it. And so the liver, the pancreas sends out insulin, the adipose tissues say, nope, we're not listening, insulin resistance. We're not going to listen to this. Next time you take a slurp, we dump out this insulin. Nope, this is even more insulin because now our blood sugar can't go down. Now we've got, no, absolutely not. We're not taking in any of this, adipose. We're not taking in any of this sugar. And so ultimately, the pancreas is yelling at the adipose tissue who's completely not listening until it just burns out. So you get this one phenomenon that cycles back and forth until it breaks down. Another example of this could be the extremely anxious person or depressed person who thinks that they're stupid. They can't study, they're thinking about they're stupid, they're thinking about how they're going to fail. They go in to take the test, they're obviously nervous, they can't think right. They get a bad grade on the test, duh, because I'm stupid. And so you get this cycling of negativity leading to negativity, pulling you into your depression. Similarly, you can think about aggression that way. If you view people as aggressive, and you attack them, and you get retaliated against, you're right, they were out to get me. So now I got to be more careful or more paranoid. And so it's these attractor cycles that we can see that one thing is spiraling out, and the idea is going to be to catch that early, if possible. And so I'll end there. I didn't get to get through all of this, but I think I got the general idea across. I'm sorry, y'all can ask questions if you want. I'm more than happy
Do you
to.
define attractor for us? So
Oh, I'm sorry. I threw this term out and it didn't mean anything. But it's the idea of two. It's probably a better word for this that people could understand would be a vicious cycle. So it's just, one thing is leading to an next, sort of a snowballing effect. A causes B and B causes A, or increases, each one increases the next. So A leads to more B leads to more A leads to even more B, that sort of thing. That's right. And that's where most of our predictive behavior comes from. When we say, past behavior is the best predictor of future behavior, that's probably only most true in things that have this type of nature to it, A to B cycling phenomenon. Any questions?
Would you mind, I'm sorry, just repeating
So, again, I'm going to talk a lot about homeostasis. In fact, I have a whole theory on homeostasis because I think it's extremely important, and I think it's what's happening. I think it's largely what's happening in most of our psychiatric conditions is that our body tries to maintain some level of homeostasis, but it is unable to do it for some reason or another. And so all that means is that there's lots of ways to modulate a system. If your body wants more serotonin, it can upregulate how much is packaged. It can downregulate how much gets broken down. It can decrease how much gets reuptake. It can increase the amount of receptors. Once that is our body's unable to sort of manage that to some extent, I believe illness gets expressed. Or we run into a catch-22. So by increasing the amount of neurotransmitters, you can help this one avenue, but you hurt this other avenue. So something that one thing is absolutely going to dictate the next two. Sort of you can't have one without the other.
These are levels of response. I mean, everything's go back down to the protein level.
Mm-hmm.
It's all a homeostatic mechanism that manifests itself to adapt or not adapt based on whether it's achieves a goal or not a goal. Although it's a different level of, one is literal, the other is actually more figurative judgment of all the project.
That's right. So all of this obviously happens within a context of environment and culture. But I don't think that it's just as simple as what's going on in the protein level. I think that it also really does require the environment because that's where it all starts. Because what I didn't say is before we get our muscle movement, we have to get a signal from the environment, whether that be, I'm hungry, my blood sugar is low, send up this, go feed, I see food, I recognize it as food, it is this far away, the cerebellum makes the calculation. It is the environment largely, whether it be the external environment or the internal environment, that's driving a lot of this. So that's sort of inherently within it. And if you change the environment, what was pathological can be not, or could be adaptive to some extent.
Zach, are you just saying, it looks like that's 5 different ways of saying the same thing?
Well, when you talk about pathological learning, you're putting the learning is an adaptive process.
Mm-hmm.
Derived from homeostatic drive. So the idea is it just sort of levels of complexity to understand an abstraction of basic biological process. Throwing more serotonin, drive the equation, a direction that creates an adaptation that requires more suggestions to do some common compressions.
So let me clarify what I mean by pathological learning, because that's not what I think I'm trying to say. What I'm trying to say is that pathological learning would be akin to a phobia. So all of our mechanisms are operating perfectly well, but for some reason, a stimulus got paired with that, let's say a camera. I see a camera. At the moment I see a camera, I get stung by a bee. But I'm at a point in my life where I'm making this association between the two. And now all of my mechanisms were there working perfectly. I took the pain, it released cortisol, I made the association with the camera. It became bad. And now, whenever I see a camera, I get scared. All of my pathways were working perfectly. It was just an untimely association. That's not necessarily a homeostasis issue. It's more an issue of just these things got paired and it wasn't adaptive. Because our brain doesn't think. Usually on the immediate level, again, neurons that fire together, wire together. The way that we go back and rewire is we, debrief, so to speak. We look at that situation and we think, well, that camera probably wasn't dangerous. It was probably a bee. Therefore, I'm now pairing B with danger, camera good, B bad. And then I'm reassociating things. So
you're saying the system could be in homeostasis, but
just because of chance, just because of random chance.
But surely that'd be an abnormality in the neural circuitry, right? I mean, if it's all anatomical and physical and chemical, as you were saying.
It wouldn't be an abnormality. It would be an maladaptive normality.
It would be perceived as an abnormality by us.
That's right. That's right. And similarly in 2, that would not be necessarily early on any abnormalities. You could not necessarily go in there and see, hey, look, serotonin is messed up here. Hey, look, this neurotransmitter is wrong. There's not necessarily a gene that's going to predict 1 or 2. Because those could be completely normal, healthy responses to an environment. Likely, they're going to lead to problems. But on the immediate level, that could have been, at that time, a perfectly normal homeostasis.
Maybe the maladaptive part becomes in being unable to learn that.
That's right. That one, but that doesn't necessarily, at that moment, imply sort of a problem in serotonin or anything like that. It's just the wiring, the hard wiring.
May have been an inability to discern the difference when the event happened. So that you can't say, oh yeah, it's a bad thing, but it's got nothing to do with Ivan Chelatop. I can see that. But you were not able to discriminate that. And so it somehow, probably because how much epinephrine and how much cortisol you got going on right at the time, because you're freaking already. So you associate everything with the flash forward. And then you can't unlearn it. So what's the drug you'd give to avoid that if you knew you were going to go into a horrible situation?
Beta blocker.
Beta blocker. Because it'll block the epinephrine and you won't get PTSD.
As an aside, you know what drug you could also give that really works? An ERK inhibitor. So ERK, as we're going to learn on the
I don't have one of those.
lecture on memory, and I can't get into your brain either. ERK can completely stop learning if you inject it in the right part, because ERK is what's going to regulate dendritic spine formation.
I don't think I want to do that.
Yeah, all of my college experience. I really appreciate I really appreciate you all asking questions.